Hello everyone, my name is Ashish Katyal. Today I will cover a topic related to bioinformatics in the era of genomics. In this particular session, I will deal about the introduction of bioinformatics. In this introduction, I will cover major things like what is bioinformatics, the historical perspective of bioinformatics. Along with that, I will cover why we need bioinformatics and how can we use bioinformatics. So, in this case, I will first tell you about what is bioinformatics. So, the name bioinformatics consists of three major things. One is bio, second is info, and third one is mathematics. So, this bio means biology, the first thing. So, it in this particular domain, it deals with life sciences. This life sciences include biomolecules. These biomolecules majorly involve five different type of biomolecule. One is known as nucleotides, which majorly involves DNA and RNA. Second is related to proteins or amino acids. The third is related to carbohydrate and lipids. Fourth is related to hormones or enzymes. And fifth is related to vitamins. So these all are not majorly used, but in this case of bioinformatics, our major role is related to nucleotide and proteins. It doesn't mean we are not dealing with hormones or maybe enzymes or maybe vitamins or maybe the energy molecules like carbohydrates and lipids. These are also important, but major focus was on this only, that is nucleotide and protein. In nucleotide also our major focus is on DNA. You can say that, sir, why RNA is not important? This RNA is also important, but in bioinformatics we usually deal RNA with cDNA. As you know that in case of central dogma there is a process known as reverse transcription in which RNA is converted into complementary DNA. So we take this particular complementary DNA from that database, and we can use that in our studies. So in case of biology, we majorly deals with biomolecules. Second is information technology, that is info, here it is written. So in this information technology, we majorly deals with database creation. In this database creation, we usually store our data. Which kind of data? Biomolecules related data. For example, if I'm creating a database related to DNA, it's a DNA database. We are representing it as like this. For example, NCBI is a popular example in which we are storing our DNA, and the repository where we are storing is GenBank, for example, it is. Or we are storing our protein molecules, or we can say protein data is there. So we can say it's a protein database. There are various examples of that. For example, Swissprot is there. For example, Uniprot is there. Various database are there. So this information technology is helpful in creating these kind of databases. So you can say that, sir, can we create a database for carbohydrates and lipids? Yes, we can create that. Or we can create a database for enzyme. Yes, we can create that. So for any kind of data, we can create a database. So how can we create the database? By the use of information technology. The third and the important part, other important part, is mathematics. In this we can use various kind of calculations. For example, in case of DNA or in case of genome of an organism, how many number of coding genes are there? You have heard about human genome project that has been completed in first draft has been completed in 2001, then subsequently in 2003 final draft has been completed in 2003 to 5. Two major, one is government organization, another is private organization, who are dealing with these particular mathematical analysis of genome. In that they have shown that there are 25 to 30,000 genes that are actively present in human genome. So how they calculated that? By using mathematics only. And along with that mathematics, they use statistics. Various calculations are done, but along with the statistics. For example, if you are saying this human genome is closely related to orangutan or maybe chimpanzee, how can we say that? By using a term that is e value, which prominently used in a tool that is known as blast, basic local alignment search tool. I will deal this particular tool in subsequent sessions, but right now I am just saying that the use of mathematics along with statistics in bioinformatics. So this bioinformatics include three different things. One is biology, information technology, and third one is mathematics. So in these three different branches, we can say that it's a combination or amalgam of three different branches of various different disciplines. So we can say that bioinformatics is a combination of three different branches, or we can say that it's an interdisciplinary branches that deals with biology, information technology, and mathematics. Same thing but different ways to say. So it's an interdisciplinary branch. After that, we should know who has coined the term bioinformatics. So there was a lady known as Pauline Hogweck from Netherlands who published a paper in her language, not in English, but in this, she has told about this bioinformatica, which has been translated into English about bioinformatics during 1970. After that, another person that is known as Jean Michel Claverie in 1980, who has proposed the term bioinformatics in his book known as Dummies for Bioinformatics, or maybe Bioinformatics for Dummies, that is written here. For example, you can check the link where he has told this kind of thing about bioinformatics. This is not that much important, but the major question is why we are studying bioinformatics. As you can see, there are a lot of data that has been come during 1970s after the discovery of a technique known as Sanger sequencing. Sanger sequencing was a method by which we can find out number of base pairs, that is number of ATGC, that are present in a nucleotide sequence. So due to that, lots of data has been come exponentially in a exponential way in a database known as NCBI. For example, the data is from NCBI only. Along with that, around 2000s, or we can say that 2001, or maybe lots of data has been come from whole genome sequencing, WGS, whole genome sequencing, or genome sequence or gene sequence are also coming, or the DNA sequence are also coming at the same time. So due to this, lots of data, exponential data has been come, and due to this, I'm showing you uh, from the original link of NCBI how they have used that particular thing for the creation of database. So this was the link which here it is present. So you can check it out, by which we can use bioinformatics for the creation of database. So next thing that comes into our mind is related to cost and time. As the lots of data has been come, for example, I'm saying that I am making a petri dish for the inoculation of microorganisms. It needs some a certain amount of money, it needs certain amount of time for doing that particular experiment. But for example, right now it is 2022 which I am doing right now this experiment. But after certain interval of time, for example after 50 years, I came into place, and I see that it needs lots of replacement of the culture so that it can retain the microorganism, and at the same time lots of time is also required for doing that experiment. So this was the major problem that was available, that needs immediate attention of a new method, or you can say a new branch that is known as bioinformatics. I will tell you how we can use bioinformatics to overcome this cost and time issue. So second problem was that, first problem was related to data, lots of data was coming. Second is related to cost and time, lots of money as well as time was needed for performing the same experiment again and again. And again means data redundancy. So for example, I am doing certain thing in India, so some other person is doing the same thing in USA, or maybe Canada, or maybe UK. Anyone is working on the same problem, but why, what is the necessity of doing same experiment again and again if we can reduce the time by the use of bioinformatics? So I will tell you how we can reduce this data redundancy or repetition of data by the use of bioinformatics. The fourth thing that came into our mind is related to protein structure. By simply looking at the protein sequence, we cannot determine its structure as well as function. For example, if you are giving me a string of amino acid, for example string of amino acid is there, it's a primary sequence, you can say it's a string of amino acid. And I'm asking you that what is the structure, secondary structure of this particular amino acid. And a random guess is that it may be helix, it may be beta sheet, it may be turn, or it may be coil kind of thing. But it is just a guess, but how much sure we are? So there is a problem in protein structure prediction, or we cannot say that this particular amino acid sequence will result it into certain kind of function. So there was a problem. So that's why bioinformatics came into picture, and I will tell you in subsequent slide how we can use bioinformatics to overcome this problem of protein structure and function prediction. The next problem was related to evolutionary relationship. This is a funny picture in which there is a organism known as human, and there is a ancestor that is known as, for example, chimpanzee or maybe gorilla is there. So how can we say that this gorilla or maybe chimpanzee is related to human? If it is related to human, how much it is related, or how much evolutionary distance it is there, or how much character difference are there, character in terms of base pairs are there in terms of human as well as chimpanzee or maybe gorilla. So how we cannot predict the evolutionary relationship by simply looking at the data. So this was a problem at that time. Next is related to gene expression. As you know, in case of central dogma, DNA is converted into DNA, that is known as replication. DNA is converted into mRNA, that is known as transcription, and mRNA is converted into amino acid, that is amino acid sequence, that is known as translation process. And as I earlier told you, there is a process known as reverse transcription in which mRNA is converted into cDNA, that is known as reverse transcription. So in this case, we can say that, for example, I'm giving you unknown query of DNA, and I'm saying that, can you tell me whether this particular function, whether this particular segment is functional or not? Can you say that? So we have to find out whether it is expressing or not. Expressing means whether it is giving a functional product or not, or it is giving a functional protein or not. So this kind of expression thing, whether it's a functional protein or not, or maybe non-functional protein, whether it is giving a product or not, this kind of thing was not easy at that time of biotechnology during 1970s. This was a problem at that time. Also, we cannot make a drug, we cannot find out the potential compound that is available in that particular protein which can be used as a drug. So this was a problem. So I have listed 6 7 problems. Based upon that, a new branch came into picture, that is known as bioinformatics. So the next question arises, how can we use bioinformatics? The first thing which I've told you in the previous slide was related to exponential data. Lots of data was coming, and you have seen that during COVID era lots of data has come, and lots of genome data has already been come. WGS was there, whole genome sequencing was there, but there was not a place where we can store this huge amount of exponential data. We cannot store in a single lab. If we are storing it, the other person will not able to use, and there were other timing constraints, timing as well as cost constraints were also there. So due to that reason, bioinformatics was come into picture, and we have created a thing that is known as database by the use of information technology. In this, we can store our data and we can retrieve our data at any time. You can see that, for example, you have seen various kind of database, for example, airline database or maybe railway database. You have seen that we can book a ticket anytime. For example, transaction database, for example, banking database, you can send money at any time or with a least amount of cost, or maybe it may be free. The service may be provided by free, by, or maybe minimal charges were there by your bank or maybe airlines or maybe railway. So this kind of thing is due to database creation. In bioinformatics also, we have created various kind of database, either it's a nucleotide database, or it's a protein database, or it's an expression database, or specialized database, or disease database, or special organism database. All kind of database has been created by bioinformatics only. So by doing that thing, it reduces the cost, it reduces the exponential growth of data. Doesn't mean exponential data is not coming, it is coming, but we are storing it. And the good part is we can store it at any time and we can retrieve it at any time. Along with that, time also reduced. Someone is doing the experiment in UK, do not have to perform the same experiment again and again, so it reduces the data redundancy also. So if I have already performed an experiment in India, so he can, if it is correct, if it is curated, then he can take the data from that particular point and do his research from that particular point. So this kind of thing was there, and that's why how we can use bioinformatics for that first three problems. That is, first one is related to exponential data, second is related to cost and time, and third one is related to data redundancy. This data redundancy can also be reduced by the use of second thing, that is known as sequence alignment. Sequence alignment is a key term in which we have to find out, for example, this is my query, and for example, unknown sequence is there. I am saying that it's a DNA sequence. Can you tell me it is related to which organism? So why I'm saying that? Because it will reduce my time as well as cost for doing the same experiment again and again. So I do not have to find which organism it belongs to. I simply have to use a tool that is known as BLAST, using sequence alignment, pairwise sequence alignment, in which I will align my query sequence with the database. And if it is matching, for example, it is matching with that database, for example, it is of human, so I can say that it belongs to human, or it belongs to maybe chimp, so I can say that it belongs to chimp. So what is the advantage of using sequence alignment in bioinformatics? In sequence alignment we can find out orthologs and paralog species. Ortholog species means, for example, if unknown sequence is given to us, or it's a new sequence which I have isolated from an organism which is unknown to anybody. So if it is related to certain database, then I can say that its function may be similar to that particular organism. Or there is a thing that is known as gene speciation and gene duplication. For example, there was an ancestor X, there is a recent species A, B, and C. For example, I am saying that these two particular things are related to each other. So it may be possible that certain genes are also possibly same in those organisms. If those genes are same in those organisms and performing the same function, probably same function, then we can say that these genes are orthologous. For example, hemoglobin coding gene in human as well as chimp, these are orthologous genes. We can also find out paralogous thing. For example, same gene performing a different function but present in same organism. For example, I'm taking another example of the same thing, that is hemoglobin. Hemoglobin coding gene in nerve cell and RBCs. So these are the examples in which same gene is performing different function. In one particular picture, it is overexpression, it is, and in another particular picture, it is underexpression. So this particular paralogous thing is due to gene duplication event. For example, there was a gene known as CCC, that is the sequence of that particular gene. It came here, for example, in the form of mutation, but in this case, it came into CCC in a normal form. For example, it is CCC in a duplicated form. So we can say that a gene duplication has been happened, and this organism is also same. For example, chimp, two different chimp species are there, so duplication event has been taken place, so we can say that it is paralogs. So we can say use sequence alignment in that particular sense also. Next is related to protein structure and function prediction. In the previous slide I have earlier told you there are four levels of protein structures. So by the use of bioinformatics we can easily determine the sequence, we can easily determine the secondary structure. Either determine means we can predict, it is not 100% correct, but based upon the algorithm we can say that this much surety is that it must consist of this much number of helix, this much number of beta sheets, or maybe turns, or maybe coils kind of things that are available. Or it may consist of certain kind of motif or functional part in the protein, or it may consist of this much number of domains, or it may consist of this kind of superfamily of that particular protein. So this kind of thing we can predict by the use of bioinformatics, or the protein structure and function prediction we can do by the use of bioinformatics. Another thing which we have told about the evolutionary relationship, as I earlier told you, I've given you certain examples based upon that. For example, this is human, this is, for example, chimp, this is mouse, for example, this is rat. These are four organisms are there, and we are saying that they are ancestrally linked to each other. For example, I'm saying this chimp and human are related to each other, this mouse and chimp are related to each other, or maybe mouse and human are related to each other, this mouse and rat are related to each other. So this kind of analysis of ancestor descendant, this is ancestor, immediate ancestor, and these are immediate descendants. These are ancestor, this is ancestor, and this is the major ancestor. For example, X is there. So this kind of ancestral descendant relationship we can find out by the use of bioinformatics under phylogenetic analysis. Also, we have told about a term that is known as gene expression. How can we find out the gene expression? That is whether the gene is on or off. So by the use of microarray technology, we can find out, for example, this is a chip in which we are using various fluorescent labeled genes as a probe. If we are using it, then for example green color comes, then we can say that the genes are on. If it is binding to this our sample DNA and it is showing certain colors, sorry, based upon that we can say that either it is on or it is off. If green color comes, then we can say that it is on, and for example if red color comes, then we can say that the gene is off. Or maybe some intermediate color are also there. By the use of this microarray technology, we can find out this gene expression thing. The last and not least thing that we can cover by the use of bioinformatics is drug discovery and designing. There is a process of drug designing and discovery that can be segregated into five components. For example, first one is known as target identification, second is known as target validation, third one is known as lead identification, fourth is known as third is known as lead identification, fourth is known as lead optimization, and fifth is known as preclinical trials. All things comes under this particular thing that is known as drug designing. So the most important thing that is known as in this particular thing is lead identification. So you have seen in the COVID era that a drug has been or a vaccine has been discovered or by the use of bioinformatics only, in which they have identified certain kind of lead compounds that can be used or that can be used for the treatment for COVID. This kind of thing comes under this drug discovery and designing. We will deal in more detail about this particular topic in the subsequent slides or maybe subsequent sessions. So right now I'm stopping here, and good luck for learning new topics about bioinformatics in this particular channel. Thank you so much.